Hello, my name is Joshua Mugabi. I welcome you to Replication, Transcription and Translation. Our lesson objectives are central dogma of molecular biology, semi-conservative DNA replication, polymerase chain reaction PCR, transcription, translation, genetic code, insulin production, or universality. You are welcome. Central dogma. The central dogma of molecular biology describes the flow of genetic information. The central dogma of molecular biology describes the flow of genetic information. DNA may be copied during the formation of new cells, and that is via DNA replication. DNA may be copied during formation of new cells via DNA replication. So we want to see here our DNA. We have DNA here, and this DNA is the one that is going to replicate. And when it replicates, it will make a copy of itself, that is during cell division. DNA may code for RNA transcripts, that is messenger RNA, via the process of transcription. The DNA, we are saying, may code for RNA transcripts, which we refer to as messenger RNA, via the process of transcription. Now, DNA, our DNA, is the genetic code. It's the one that has the code. So what happens is that during transcription, it codes for RNA transcript, and it forms what we call the messenger RNA. So the messenger RNA is the one that transports the information that will result in the production of our proteins. The messenger RNA transcripts codes for the production of protein via translation. They code for the production of proteins via translation. So when we look at this structure once again, we shall see that during DNA replication, what happens is that the DNA molecule is going to be copied. And when it is copied, it will pass over the information to the messenger RNA. And that process of passing over the information to the messenger RNA is what we refer to as a transcription. That is transcription. Transcription is the passing over the information from DNA to messenger RNA. Then the messenger RNA will take the information in the cytoplasm to the ribosome, where the information is going to be translated. The information will be translated, and then we shall end up forming our protein. We shall form our protein that we have got the information from the DNA. So that's why we are saying that messenger RNA transcripts, they code for the production of proteins via translation. Let's look at comparison of the cell processes. And which cell processes are we talking about? We are talking about DNA replication. There is translation, transcription. We want to compare those processes. Now we shall look at the points where they occur, the location, when it occurs, key components, and the functions of those different processes. We have DNA. DNA occurs in the S phase, and transcription, it is always continuous. Then translation is also always continuous. The location of DNA replication, it occurs in the nucleus. Transcription also occurs in the nucleus. And then translation occurs in the cytoplasm. Then we have the key components. The key components in DNA replication, we have the enzymes. And then we have transcription. They are the enzymes still. And then we have translation. Ribosomes are the ones that are involved in translation. What are the functions of DNA replication? It duplicates the genetic information that is prior to cell division. And then transcription. This one produces RNA transcripts, or what we call messenger RNA, of the DNA sequence. And then translation makes the protein according to RNA transcripts. So those are the comparison of the cell process: DNA replication, transcription, and translation. Let's look at replication in detail. We have the semi-conservative. DNA replication is semi-conservative because when new DNA molecules form, they are composed of one strand from the original template and one newly synthesized strand. So let us look at this semi-conservative and we'll try to understand it. Now we have our DNA. This is our DNA, and this DNA is the one that is going to give us the template strands, the one having the template strands. So we shall look at how replication takes place, or unwinding, opening, and so on. So what happens is that the DNA double helix will open, and when it opens, now a new strand will be formed. There will be a new strand formed along one side, and then another strand will be formed on this strand this way. So when those strands are formed, now we shall get two newly synthesized DNA, meaning that the DNA has replicated itself. But now when you look at this DNA, you are going to see that it has the blue structure, which is from the original. It has the blue structure, which is from the original, and it has a strand which is red, which is the new synthesized structure. Now, for that reason, that's why it is called a semi-conservative, because the new formed products, you will see that they have what we call the original. They have the original template, the one which is blue is the original, and one newly synthesized. The red one is the newly synthesized. So a new chromosome or a new DNA will have both the old strand, one of the original old strand, and one of the newly synthesized strand. That's why it is called a semi-conservative. This occurs because each nitrogenous base can only pair with its complementary partner. For example, adenine combines with thymine. and then guanine combines with cytosine, meaning the base sequence is conserved. The base sequence is conserved during replication. So here what we are looking at, we are saying that the semi-conservative, this one occurs because the nitrogen bases can only pair with its complementarily. It can only pair with its complementarily. For example, if you look at adenine, adenine can only pair up with thiamine because they use a double bond. They use a double bond. Whereas guanine can only pair with cytosine because they use a triple bond. So they are complementary. They complement each other. And that's how they pair up. Therefore, the base sequence is conserved. The base sequence is conserved. So you will find that this DNA here we have here, this DNA here we have is going to be the same DNA as this one because they have the same bases which have combined. Melson-Stahel experiment. Melson-Stahel's experiment supported the semi-conservative replication theory. They gave two radioactive isotopes of nitrogen. These ones were introduced during the replication process. The DNA molecule was first incorporated with a heavier nitrogen-15 isotope. Then DNA was induced to replicate in the presence of a lighter nitrogen-14 isotope. DNA was collected via centrifugation after division and isotopes content assessed. After the first division, DNA contains both nitrogen-15 and nitrogen-14. Now this one disproving the conservative model. After the second division, DNA consisted solely of nitrogen-14. And this one disproved that is passive model. Evidence for the semi-conservative model. Now let us look at it here. We say that there was the first division and then the second division. In the first division, they were provided with nitrogen-15. Nitrogen-15, a heavy nitrogen-15 was provided. That is during the first division. In the second division, they provided nitrogen-14. So during the time when they provided the first division, because the original DNA had nitrogen-14, now they provided it with nitrogen-15. So after the replication, it was found out that there was a mixture in the new chromosomes which were formed. There was chromosomes with nitrogen-14 combined with nitrogen-15. That was the actual observation here. Nitrogen-15 was combined with nitrogen-14. That is during the first division when they provided the heavy nitrogen-15. Now during the second division, they provided the normal nitrogen, nitrogen 14. And the result, what was obtained, some of the DNA had nitrogen 14 only. And the other DNA, they had both nitrogen 15 and nitrogen 14. Those were the actual observation. Now then we come to these other predictions of the conservative, semi-conservative, and the dispersive. For the conservative, during the first division, replication, the conservatives say that for them they say, we shall get chromosomes which are only having nitrogen 14 and other chromosomes will have nitrogen 15 only. The semi-conservative, for them they say, we shall have both chromosomes or the chromosome mixed up with nitrogen 14 mixed with nitrogen 15. Then the dispersive, for them they say, yes, we shall have chromosomes with mixture of nitrogen 15 and nitrogen 14. So you find out that here, the correct observation, actual observation, were corresponding with those ones of semi-conservative, and they also corresponded with those ones of dispersive. They did not correspond with those ones of conservative. Now, when they went to the second division, in the second division, here they provided now, they changed and provided nitrogen 14. And the actual observation was that the results they obtained, they were some DNA which had only nitrogen 14. And there were other DNA that had a mixture of nitrogen 15 and nitrogen 14. So, what was the prediction of the conservatives? For the conservative, for them they said, here, where you provided nitrogen 14 in the second division, we would get only chromosome with nitrogen 14 and others will only have nitrogen 15. What was the prediction for the semi-conservative? Semi-conservative, they said, we shall have nitrogen 14 chromosomes and we shall have chromosome with both nitrogen 14 and nitrogen 15. And what was the dispersive prediction? Dispersive prediction, for them they said, we shall have a mixture of nitrogen 15 and nitrogen 14. Those are the only ones that we shall have. So here you find that the only prediction which corresponded to the actual observation was that one of the semi-conservative. The one for dispersive, this one they failed because for them they sticked to having only a mixture and no chromosomes with only nitrogen 14. So this one failed. And the one for the conservative, for them they said the only thing they can get will be only nitrogen 14 chromosome only and nitrogen 15 chromosomes only, which was also wrong. So there, now we came up with the conclusion that this replication of DNA is a semi-conservative process, not conservative and not dispersive. DNA replication. DNA replication. The purpose of replication, is to duplicate the DNA to create identical sister chromatid prior cell division. The purpose of replication is to duplicate the DNA to create identical sister chromatids prior to cell division. So before cell division, before replication, we have one copy of DNA. We have one copy of DNA. Now this one copy of DNA, it undergoes what we call replication. Replication, it is going to duplicate itself. And after duplicating, now we get what we call the identical sister chromatids. So after duplication, this is what we get. We have now two sister chromatids. These ones we call them two sister chromatids. Now when you look at them, they are two chromosomes that have been formed or that have been replicated, that have been duplicated. So here we have two copies after the process of replication. So the purpose of replication is just to duplicate the DNA to create sister chromatid before we go to cell division. The enzyme helicase, separates the DNA strand. The enzyme helicase is the one that is going to come and separate the DNA strand. It will separate the DNA strand into two so that we can carry out the replication. Then the enzyme DNA polymerase. We have the enzyme DNA polymerase is the one that is going to copy the new strand. It's the one which is going to copy the new strand. When the DNA molecule or strand opens, then we get an enzyme which we refer to as DNA polymerase, and it copies the DNA, and then that one can be used for replication. DNA replication. So we are saying that we have the helicase. Helicase is the one that winds or opens the DNA molecule. And then we have DNA polymerase is the one that is going to form other strands from the template strand. So here we can see that now, right now, that is helicase. It is going to unwind. It will unwind the DNA. Then now polymerase will come and then put there the nucleotides, and then we form other chromosomes. Helicase. Helicase enzyme, the helicase enzyme, this one unwinds. It unwinds and separates the two strands of DNA double helix. That is the purpose of enzyme helicase. Enzyme helicase, for it, its work is to open, to make sure that the DNA double helix is opened by breaking the hydrogen bonds. It breaks those hydrogen bonds. And when it goes on breaking the hydrogen bonds, therefore, the templates will separate. One goes up, another one goes down. And now that means that we are exposing the complementary bases. The complementary bases have been exposed so that new DNA molecules can be formed. The template strand can be copied to make two sets of identical DNA. So we can copy the template strand. The strand that is going to be copied is what we call the template strand. They can copy this one here, and when they copy, they make other chromosome. And if it also copies this side here, then we shall get to DNA molecules. So that's what we are saying. The template strand can be copied to make two sets of identical DNA molecules. So the DNA that we get this side up after replication, and the ones that we get down this side after replication, all those ones are called sister chromatids or DNA, identical DNA. DNA polymerase, DNA polymerase. Helicase, the helicase, for it, what it does, it separates the strands. And after separating the strands, the free nucleotides will line up opposite to the exposed complementary bases patterns. And that is via hydrogen bonding. Now I want us to look here very carefully. We have this one is our helicase enzyme. For it, its work is to separate the DNA strand. After it has separated the strands, now we are going to have another enzyme, which is called DNA polymerase 3. Now this one is going to be involved in now the combining, aligning of the complementary bases. So when it is moving, the complementary bases will be coming and joining, lining up with other complementary bases. And as they come and combine up with other complementary bases, that means that now we shall have what we are forming behind it. Behind we shall be forming another strand, DNA strand. And this one, for example, when you look at this one is polymerase, DNA polymerase 3, which is responsible for bringing the nucleotides to line up. This is our nucleotides. As these nucleotides, they come and line up the bases, complementary bases. These bases, as they come and line up here, they will combine with these bases. And as they combine with those bases, now we shall form another strand behind, another strand of DNA will be formed behind. But always when they are building, they go in the direction of 5 to 3. And this side also, this one will go in the direction of 5 to 3 as they are creating the new DNA strands. So DNA polymerase 3, this DNA polymerase 3 covalently joins DNA nucleotides together to form a new strand. So this is our polymerase, DNA polymerase 3. It is covalently joining the DNA nucleotides. These nucleotides, it joins them together and it forms a new strand. We form this new strand here. This is another new strand of DNA that has been formed. Now when you look also at this DNA polymerase 3, it will bring these bases or nucleotides, combine them together, and behind it we shall form a new strand of DNA. DNA polymerase 3 synthesizes new strands from the template strand. So this one in red, the one in red is the new strand. The other one, the one in blue, that is what we are calling the template strand or the original strand. This one in red is the new strand. The one in blue is the original strand. and covalently joins the nucleotide together. So we are saying the purpose of DNA polymerase is to form new strands, and then also to covalently joins the nucleotide together. This new strand that we have formed, these new strands that we have formed, they are made up of nucleotides, and the nucleotides have covalently joined each other. That's why we are having now a new strand, because the nucleotides have joined covalently together. DNA polymerase links the nucleotides together to form new strands. And this one, it uses the pre-existing strand as a template. So this is our DNA polymerase. DNA polymerase is going to link the nucleotide together. It has linked the nucleotides together, and we have formed a new strand, which is red. And it is coming from the pre-existing strand. This is the blue one, which is the pre-existing strand. That is the pre-existing strand. But now, after polymerase linking the nucleotides together, they formed a new strand, which is red. Polymerization. The new nucleotides align opposite their complementary pattern as deoxyribonucleoside triphosphate. So when you look here, you will see that this is a nucleotide here, and this one is a nucleotide. And here we are having our DNA polymerase 3. So it is the one which is combining these nucleotides. It combines this nucleotide to the other nucleotide. So it is the work of DNA polymerase to join the nucleotides together. So you can see that's how it is joining. You can see that a new nucleotide will come on the DNA polymerase, and when it comes, it will form a bond, and the nucleotide will go on increasing, and when we are getting a new DNA molecule. When joined to a growing chain, DNA polymerase will cleave two of the phosphates, thus releasing energy. So when you are seeing here, the nucleotide comes with 3 phosphates, and 2 are cleaved off. Those ones, when they are cleaved off, they are the ones that produce energy, which is used to make the bond. That's why we are saying, when joined in a growing chain, DNA polymerase will cleave 2 of the phosphates. And when 2 of the phosphates are cleaved off, this one releases energy. And of course, the energy is the one which is going to be used to make bonds between the nucleotides. The energy is used by the enzyme to create covalent phosphodiester bond. It is used by the enzyme is DNA polymerase, to create covalent phosphodiester bond with the nucleotide chain. Of course, the one we have up this side is the chain, And these ones which are coming from down, these are nucleotides. So when they come to the enzyme what it is going to do, because it is getting energy, it is going now to combine the nucleotide to the long chain of the nucleotide, forming a bond which is a covalent phosphodiester bond with the nucleotide chain. The synthesis occurs in the 5-3 direction on a new strand. The synthesis occurs in the direction of 5-3 on a new strand. So when they are synthesized, they are moving from carbon atom 5 going to carbon atom 3. Polymerase chain reaction. Polymerase chain reaction. The polymerase chain reaction is an artificial method of replication used to rapidly amplify DNA sequence. Now the other ones are occurring in the cell. The other replication is taking place in the cell, but this one can be induced artificially. The replication of DNA can be induced artificially, and this one is mainly for amplifying the production of DNA sequence very rapidly. What do they do? 1. Polymerase chain reaction involves 3 key steps, which are repeated. The first one is denaturation. Denaturation. Denaturation, here, heat separates the strand. They use heat to separate the strand. Remember, this one is artificially carried out. It is not carried out inside the cell, whereby we have the enzyme helicase, which is going to open. But now this one, it is artificially, which means there is no helicase. And for that reason, to separate the strand, they are going to use heat. Heat is the one which is going to separate the strand. Here we have our strand, which is still bound. Now, when they increase the temperature to 90 degrees, 95 degrees Celsius, then you can see that now our strand is opened, or it has been wound up. So, annealing. We have annealing. Primers designate copying sequence. The primers designate copying sequence. Now here they reduce the temperature to 55. When they reduce the temperature to 55, now the primers will come. These are the primers. They will come and start the replication process. And they start copying. Then the third one is elongation. This is where the tag polymerase copies the sequence. The tag polymerase copies the sequence. Now you can see here we have another strand formed down and another strand here formed up. All of them, they have just made a copy of the DNA. Polymerase chain reaction of 30 cycles generate approximate or greater than 1 billion copies of DNA. So you can see that polymerase chain reaction can produce a lot of copies of DNA. Use of the tag polymerase produces a multiple copies of DNA rapidly by polymerase chain reaction. This one is the artificial method of preparing or making copies of DNA. It is cheap and it is fast. Let's talk about transcription. 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 The purpose of transcription is to synthesize an RNA sequence from DNA template, and only one strand is copied. So the purpose of transcription, we are going to make what we are calling the RNA sequence, and this one is going to come from the DNA template. Now, when we are forming this RNA sequence, sometimes it's what we call the messenger RNA. We are copying information from the DNA template and then transferring it to the messenger RNA to carry that information for the production of the proteins. The enzyme RNA polymerase separates the DNA strand and synthesizes an RNA copy. Here we don't use helicase. Helicase, we just use it during replication, making copies of DNA. But in transcription, we are copying templates DNA. We are copying or synthesizing RNA sequence from DNA template. We are not making copies of DNA. So there is a difference between replication and transcription. Here in transcription, we are using RNA polymerase to separate the DNA strand and synthesize an RNA copy. After transcription is complete, the RNA sequence is released and the DNA strand reforms a double helix. Now, when they have finished copying the information from the DNA or coding and forming the RNA sequence or the messenger RNA is going to be released, and the DNA strand will reform a double helix. The RNA transcript, the one that we refer to as messenger RNA, then will move to the cytoplasm to facilitate the translation. Now, when the messenger RNA or RNA transcript has been got, it is going to go to the cytoplasm, where now another process which is called translation is going to take place. So here we are having 3 RNA that are going to take part. We have the messenger RNA, the one which is getting the information from the DNA. We shall have the transfer RNA, which is going to bring the complementary bases, and then we shall have the ribosome RNA. Now, here we have RNA polymerase. We have the DNA strand, and then we are forming the RNA sequence or the messenger RNA. So let us see what is happening. RNA polymerase is going to unwind the DNA, and when it unwinds, then again it will create the messenger RNA. So the messenger RNA will be formed, and after forming messenger RNA, then the DNA will again wind up again. So we have our messenger, that is RNA polymerase, is going to unwind the DNA. Now it is going to combine the nucleotides to form the RNA, which is now released, and it goes to the cytoplasm. and the DNA winds up again. RNA polymerase. Now, transcription is mediated by RNA polymerase, which performs the following. It binds DNA and separates the strand, that is RNA polymerase, therefore breaking the hydrogen bonds between the base pair. So we have our enzyme here. This enzyme here is the one that we are calling RNA polymerase. So when it comes, it will open the DNA. It separates the DNA. And after separating the DNA, now it will start creating the messenger RNA, this one inside. To start creating the messenger RNA, but as it is moving along the DNA, it is breaking the hydrogen bondings, and then behind it, it is creating the RNA transcript. Covalently joins RNA nucleotides together to form a new strand, uracil instead of thiamine. So this one, which is RNA polymerase, in front it is opening the DNA. Behind, it has already opened the DNA, and at the same time it is creating the RNA transcript by joining the RNA nucleotides together, and it is forming a new strand which we are referring to as the RNA transcript, or in most cases we call it messenger RNA. The transcription is the synthesis of messenger RNA. That is transcription. The synthesis of messenger RNA copied from DNA bases. This one is the messenger RNA which they are forming, and it is copying information or base sequences from the DNA. The DNA is the one which is outside, and this one they are copying is the one that we are calling the template, and this one is done by the messenger RNA, or RNA polymerase. Transcription is the synthesis of messenger RNA copied from DNA sequenced by RNA polymerase. Sense versus antisense strand. Sense versus antisense strand. While gene sequences can exist on either DNA strand, RNA polymerase will only transcribe the strand with appropriate nucleotides, sequence, or what we call the coding region. The antisense strand is transcribed. Sequences is complementary to the RNA transcript. So behind we have seen the DNA being unwound, or opened by RNA polymerase. Now, when it is opened, there are two opened strands, the lower strand and the upper strand. One of the strands is going to be the sense strand, and the sense strand is the one which is not going to be transcribed, is the one which is not going to be copied. The antisense strand is the one which is transcribed, and it's the one which is copied, where the sequences are complementary to the RNA transcript. The sense strand is not transcribed. It will not be copied. Sequence. It is identical to transcript. It is identical to the transcript except for thiamine, which is replaced by uracil. Now the sense strand, the one which they are not transcribing, it is the same as this new one which is being formed, which we are calling the RNA strand or transcript. It is the same as the sense strand, which is not copied. So that's the reason why it is not transcribed. For example, if we have the antisense, it has the following bases: thiamine, arginine, cytosine, guanine, thiamine, cytosine, adenine, guanine. If that is the antisense, these are the codes on the antisense. Then the sense will be having the following. The antisense has that one strand, the sense will have these ones. But what is copied is the antisense. So when we copy the antisense here, we make another copy here, a new copy, you will find that they are going to form bases here, there, here, there, 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 there. So you will find that these bases which are on this new formed one, they are the same as those ones of the sense strand. So when we form the new strand, this strand we are forming, the new strand is going to be identical to the sense strand. They will be the same as the sense strand. Only that instead of UT, here we shall have U. On the new strand we shall have U instead of having T. Then for the transcript will be adenine, uracil, guanine. So you can see that the transcript resembles the sense, apart from only uracil, which is representing thiamine. But if the transcript is CA, you will see that the sense is also cyanine, cytosine, adenine. Whereas this one is the one that we have copied up this side. So here we would get C, and A, which is this one, and which is also this one. Now we want to go to translation. Translation. What is translation? Now translation involves polypeptide synthesis. We are going to synthesize proteins. It involves the polypeptide synthesis by the ribosome. We are going to translate the information we have got from the DNA. We are going to translate it, and then we build the protein or protein production. So the polypeptide are encoded by genes and translated from messenger RNA sequence, which was produced via transcription. Remember when we were in the nucleus in DNA, there was a transcription whereby information was got from the DNA and transferred to the messenger RNA. Now when the messenger RNA moves into the cytoplasm, it goes to the ribosome, where now it is going to pass over the information to produce polypeptides. And now that process is what we are calling translated or translation. We are translating the information from where we got it from DNA. Now we are translating that information from codes to proteins. That one is done by messenger RNA on the ribosome. Transcription and translation are linked processes in protein production. So transcription comes first. We get the information from DNA, and translation, the information now is got from messenger RNA, and then we produce the protein. So the process, all of it is linked up. Synthesis of polypeptide ribosome. Let us look here and we see what is taking place. Now, when you look at this one, you can see now this is the ribosome. Then the transfer RNA will bring the amino acids. Then the amino acids will build up and then form the polypeptide via the peptide bonds. And the polypeptide will be formed. The one that we are seeing down here, this line down, is the messenger RNA. It is the one with the codes. And now the ribosome will come and align on the messenger RNA. And then the transfer RNA will come to the ribosome. They come with amino acids. And then the amino acids are going to be covalently bonded to form the polypeptide, which is going to peel off from the transfer RNA and form a protein or a polypeptide. Process of translation. Messenger RNA, we have the messenger RNA is transported to the ribosome to read the sequence in the base triplets that is called the codon. So here, when you look here, we have our messenger RNA here. That is our messenger RNA. Now the messenger RNA has the codons. The codons, these are the bases it has got from DNA. Now this one here outside, this is our ribosome. The ribosome is the one which is going to mediate the translation of the codon. Now we shall have another ribosome, this one outside here. This is the transfer RNA. That is a transfer RNA or tRNA. It is now the one which is going to bring the amino acids. And at the tip, it has what we call the codons. It has got what we call the anticodons. The anticodons are the ones that are going to pair up with the codons on the messenger RNA. And when it is coming with the amino acid. So when they come here, they will pair up. And when they reach here, they will release their amino acid. The amino acid will be combined with this polypeptide, and they will continue. It will continue growing as more transfer RNA come, and they leave their bases here, and they will be covalently bonded here. And at the end, we shall have a polypeptide formed. Transfer RNA molecules. The transfer RNA molecules, these are the ones that transfer RNA molecules here. This one is a transfer RNA molecule. This one is a transfer RNA molecule. This one is a transfer RNA molecule. They carry specific amino acid. Now here up, it is the amino acid. Those are the amino acid being carried by the transfer RNA, and will align opposite a specific codon. Then when they come, they will come and align on the codons here. The codons are on messenger RNA. They align themselves on the codon like this one. It aligns itself on those complementary codon, and now here it brings its amino acid and joins it to the other polypeptide, and they form the bonds according to the complementary anticodon. So we are saying the transfer RNA molecule carries a specific amino acid. This one here is coming with a specific amino acid, and it will align itself opposite the specific codon. It comes and aligns here on the next codon. After aligning itself on the codons here, now those ones, the anticodons, will align themselves on the complementary codons, and then they will pass over the amino acids to be joined to form polypeptides. The ribosome moves along the messenger RNA. The ribosome is going to move along the messenger RNA. You can see it is moving. It will move in this direction on the messenger RNA, and wherever it is moving, it is combining. The polypeptides are being formed. But again, the transfer RNA comes in, and it is facilitated to pass over the amino acids. So the ribosome moves along the messenger RNA and join the amino acids together. When it is passing, it is going to join these amino acids together with a peptide bond. It joins them together with a peptide bond. The peptide bond is that bond between each amino acid. Whichever amino acid they bring here combines with it to form a polypeptide. The bond between the amino acid and amino acid, that bond is what we call the peptide bond. The codons, we say these are codons. They are 3 bases. Codon is made up of 3 bases. Every codon is made up of 3 bases. So when you come and see here on your codon, you will see they are bases, and there are 3 of them. 1, let's count them here. We have this is 1, this is 2, and these ones are 3. Those ones are what we are calling the bases. And 3 of the bases, they form what we call a codon. Codons of 3 bases on messenger RNA, corresponds to 1 amino acid. They correspond to 1 amino acid in the polypeptide. So you are looking here on the messenger RNA with the codon. But again, if you look at this transfer RNA, this transfer RNA also has got what we are calling the anticodon here. They are also 3. So when it brings them here, it combines them with the codon which are on the messenger RNA and the anticodon which are on the transfer RNA. They will combine here and they will be complementary. Now, when they are complementary, at the tip, they have amino acids. Now it comes and brings this amino acid and joins it to the long chain of the polypeptides, and then it will be bonded there. When it is bonded there with a bond, peptide bond, now it will release the amino acid and it will go out to carry other amino acids to bring them for combining. Translation depends on contemporary bases. Translation depends on contemporary bases, pairing between codons, that is messenger RNA, and the anticodons. So our translation is going to depend on the codons on the messenger RNA and the anticodons on the transfer RNA. If they are complementary, then they will combine. and the amino acid which is held by the transfer RNA will now be joined to the polypeptide in the formation of the protein or the polypeptide. Translation overview. Messenger RNA goes to the ribosome, which reads the sequence in the codons, which are complementary to anticodons, which are found on transfer RNA, which carry specific amino acids, which are joined by peptide bonds to form polypeptides, i.e. proteins. Let's look at the genetic code. The genetic code is the rule by which messenger RNA sequence are converted into proteins. The genetic code is the rule by which messenger RNA sequence are converted into proteins. The codons are triplets of bases. Codons are triplets of bases, which correspond to a particular amino acid. The codons are found on the messenger RNA, and every 3 bases, they correspond to a particular amino acid. So we are calling them triplet of bases, or what we are referring to as the codons. They correspond to a particular amino acid. Now, the order of the codons determines the amino acid sequence of a polypeptide. So the way the codons are arranged, they are the ones that are going to determine how the amino acids are going to be arranged in a polypeptide. Key features of a genetic code include 1. Universality. All organisms, they use the same code. All organisms, they use the same code. Those are the key features of a genetic code, that all organisms use the same code. There are only few virus exceptions. Only few virus exceptions. Second, degenerance. Degenerance. That is, more than one codon may code for the same amino acid. We can have very many codons coding for the same amino acid. That is what we are calling degenerance. It is a feature of genetic code. More than one codon may code for the same amino acid. Because there are 46 codon combinations, but only 20 amino acids, we have 24 codons, and then we are having only 20 amino acids we use. Remember, every codon is made up of 3 bases. Every codon is made up of 3 bases. And we are having 64 codons. But each codon represents an amino acid. If you have 64 codons, how many amino acids are those? They should be 64. But how many amino acids do we have? We have 20 amino acids. So, do we need all those codons? Therefore, that means that some codons are going to code for the same amino acid. This allows the occurrence of silent mutation. This allows the occurrence of silence mutation. The amino acid sequence of polypeptide is determined by messenger RNA according to the genetic code. The amino acid sequence of the polypeptide is determined by messenger RNA according to the genetic code. Now, how the amino acids are going to arrange themselves in the polypeptide is going to be determined by the sequence, how it was, the codons that are on messenger RNA, according to the genetic code which was copied from DNA. According sequence of messenger RNA, initial with a start codon AUG. Initiating that one, now we are according sequence of messenger RNA. When coding sequence of messenger RNA, it is initiated by the codon which we call the start codon, and that is AUG. Then it is terminated with a stop codon. Degenerance occurs in the third base position. Degenerance occurs in the third base position. Degenerance occurs in the third base position. Okay, now let's look here. Wobble hypothesis. The shape of transfer RNA is not optimal for base pairing on the third position. So here when you look, these are anticodon. These are anticodon. Here, they are anticodon. And these ones down, they are codon. So the codons, they are on messenger RNA. Anticodons are on transfer RNA. But here they are saying that according to the shape of transfer RNA, it is not optimal for the base pairing at the third position. That at the third position, it is not optimal for pairing at the third position, meaning this codon here, that codon here, and codon here may not be optimal for base pairing. This anticodon here may not be optimal for base pairing at that third position. Use a table of genetic code to deduce which codons correspond to which amino acids. So here in our table we have the different codons and the specific amino acids they code for. For example, we have phenyl. Phenyl is coded by UUU, UUC, and then leucine is coded by UUA, UUG. When you go to again leucine, here you can see leucine here is coded by UUA, UUG. Again, leucine can be coded by CUU, CUC, CUA, CUG, meaning that the same amino acid is being coded by many codons. These are our codons. The combinations are our codons. So that's what we are saying. We have some of them that we start with. AUG methyl is a start codon. Then we have here UAA, UAG is for stop codon. UGA is also for stop codon. The others, they are coding for a particular amino acid. You look at arginine. Arginine is coded by CGU, CGC, CGA, CGG. All those codons are coding for one amino acid. Reading frames. Messenger RNA transcript is organized into triplets of nucleotide bases, and these are called codons. This means that 3 different reading frames exist for any given sequence. An open reading frame starts with AUG and continues until a stop codon. So let us read this one. Reading frame can be interpreted by frame shift mutation, insertion and deletion. We have messenger RNA is there. GUA, UA, GCA, G, and so on. So what do you think the codons are? What are the codons? So we start with AUG. GU is not considered because the start codon always is AUG. So we shall start with AUG. And each codon is having 3 bases. 3 bases. So can we go back to the table and we try to find out which amino acid we are going to form here? So we shall form methone. Methone is the one which is AUG, the start codon, then histamine, valine, threshold, phenyl, leucine, methyl, and also on, and then stop codons, UGA. So that's how we read the frames and be interpreted. So if the read frame can be interrupted by frame shift mutation, for example, if mutation comes and it deletes out, let's say this one, then that means that this amino acid will not be coded for, and that one may lead to mutation. Use the table of the genetic code to deduce the sequence of amino acids coded by short messenger RNA strand. Universality of genetic code. The genetic code is universal. Almost every organism uses the exact same code. That is the universality of genetic code. That almost all organisms, they use the exact same code. This means genetic information can be transferred between 2 different species. Genetic information can be transferred between 2 different species. Universality of the genetic code. This demonstrates by mass production, this is demonstrated by the mass production of human insulin by bacteria cells. So here you can see we have a human cell. We have got here a human cell. Now this human cell, and down here we have the bacteria cell. Here we get the DNA from a bacteria. And here we get an insulin gene from a human cell. Now we combine the insulin gene from human, we combine it with the DNA of the bacteria, recombinant plasmid. And then it is put in a transgenic bacteria. When it is put in the transgenic bacteria, now it is going now to be put, the transgenic bacteria will be put in the growth culture. And here it will produce our insulin, which is going to be corrected and this one now, the insulin will be given to people with diabetes. So you can see that the bacteria DNA has been forced to make insulin by inserting the human insulin gene into its DNA to form a recombinant plasmid, which is transferred to a transgenic bacteria that grows and produces the insulin. Hello, thank you very much for participating in replication, transcription, and translation. Our objectives were the central dogma of molecular biology. We looked at the semi-conservative DNA replication. We looked at polymerase chain reaction, PCR. We looked at transcription. We looked at translation. We looked at Watson and Crick. We looked at insulin production or universality. Thank you. Don't forget to subscribe.